So hello, everyone. My name is Ariana Gonzalez. I'm speaking in on behalf of the Heritas team, and I'll be talking a little bit about optimizing CNV search through Mastermind. In first place, I want to talk about who we are. We are a company focused on human health and wellness. We utilize genomic and precision medicine. We're currently based in Argentina with LATAM reach. What do? We have two units, a health unit and a wellness unit. Within our health unit, we use DNA testing to diagnose or predict risk for specific conditions like genetic disorders, rare diseases, cancer, and prenatal screening. And within our wellness unit, we also use DNA testing to obtain information that can be customized in wellness plans. So Heritas offers an end-to-end model for genetic testing. Within the health unit, we have 100% integrated solutions from end to end. In our core, we have the clinical genomic diagnostic assays. In the first step, we have the pre-analytical phase with genetic counseling. And here we have Dream24, that is our patient support tool. We offer genetic counseling and it's carried out to inform, educate, and support individuals and families. In the second step, we have raw data and interpretation phase with the genetic analysts. And in here, I want to highlight that we are an interdisciplinary team that is conformed by the platform of data production, data team or data science, and the medical genetic platform that works together in order to reach excellence in our variant analysis and interpretation. So for us, each individual is unique and we understand it as a unique way, and that makes us a better understanding than for each case. And in a post-analytical phase, we have genetic counseling, that is the interpretation phase, that is made by a genetic specialist. Since every step is consolidated within Heritas, within our laboratory, we generate and save the electronic health records that's product of the teamwork I previously shown. So this is our Heritas portfolio. We have different products. We have Vision, that's our non-invasive prenatal testing product. Focus, that is our clinical exome solution. Clear, that is our hereditary cancer solution. CHROMO, that is our microarray solution. And OncoSense, that is our liquid biopsy solution. We count with 4 very important international quality controls, such as CAP, the European Monitor Quality Network, the UK National External Quality Assessment Service, and Seracare. And this is a picture of Heritas today, and without this working team, nothing would be possible. So I want to start with use case 1. Use case 1 is from CHROMO, our array solution, and it's about a large duplication. We had a 1-year-old male patient under study for hypotonia, psychomotor retardation, macrocephaly, polycystic kidney disease, and ventricular septal defect. The high-resolution cytogenetics report reported an inversion of the Y chromosome. We performed a chromosomal microarray analysis, and the result was a 25.3 megabases distal duplication in the chromosomal region in the long arm of chromosome 1, more specifically in the last part. So as it affected the terminal region of the chromosome 1, and this region was big and contained a large number of genes, we didn't hesitate in jumping directly through Mastermind. We use the cytogenetic band, but it's important also to highlight that you could use the genomic coordinates for your search. We could easily and quickly obtain an exact overlap with our region. And it's interesting to see that also we can see regions that are contained intersecting with this region. And you can also go and tap the genes to see and compare and contrast if the genes you found in your region are the same that are in this region that you're matching with. So we found 49 articles, and since duplications that involve this region are extremely rare, we wanted to reduce this number a little. So we used the phenotype filter, and we filtered by microcephaly, and we found 7 articles. We found this article that was really interesting that talked about the pure duplications of the terminal regions and the delineations for allele trisomy. And this was interesting because it provided us vital information of the lack of actual data we have to really define the syndrome. And then we found this paper that it's from 2016 and talked about the detailed analysis of 26 cases of duplications and triplication syndromes. And this paper was really a breakthrough for this patient, because in general, carriers for these duplications are described as product of unbalanced translocations. So this really was a paper that helped us better understand the variant. It's also interesting to go and explore annotations and also see smaller or bigger CNVs that are not the one you're seeing as the highlighted, because you can always get more clinical data from them. So after issuing the report, the doctor confirmed that the karyotype delivery carried out on the patient was not really an inversion, but an addition material that was attached. And karyotype was also performed on the patient's parents, and a normal result was obtained. So we can confirm that our duplication was de novo. And we were able to find information quickly and accurately in relation to our variant and relation with the phenotype, and together with our algorithms and based on the ACMG guidelines, we classify this variant as pathogenic with no conflict. And the second case, it's a case that came from CLEAR from our hereditary cancer solution. And here is an exome resolution level example of how to use Mastermind. It's a 28-year-old male patient with a diagnosis of cerebral hemangioblastoma. We didn't have any additional medical history or family history of cancer. So the doctor was suspicious of a possible von Hippel-Lindau syndrome. The VHL gene was analyzed, and we detected a possible deletion in heterozygosity in exon 2. And the deletion was actually confirmed by MLPA analysis. So although our case was on course and ongoing, we wanted to really understand how frequent or how rare this variant was. So we searched directly for the deletion in Mastermind, and quickly and with no problem found the exact overlap with the region. And again, 86 articles were a lot to review. So we used the phenotype filter and filtered by cerebral hemangioblastoma and found 13 very interesting articles. But the one we wanted to highlight was this one that talks about the characterization of the VHL promoter variants in patients suspected of von Hippel-Lindau. And it talked a little bit about the frequencies of the different variants within the VHL gene. And only 11% corresponded to large or complete deletions. On top of that, only 17% corresponded to exon 2. So this really highlights the fact that sharing data and performing case studies is key or crucial to obtain a better understanding of variants and of the syndrome. So what are the advantages of using Mastermind? It enhances variant analysis on our daily basis. Using filters to direct searches, it's fast and easy to use, it's intuitive, and it helps in time management. And I want to stop here a few seconds because at the end of the day, it's not about delivering the report, but it's about delivering the best report possible for each patient. So for this, Mastermind plays a key role in our daily basis in order to fulfill this statement. Thank you.